Hello everyone and welcome to my channel Easy Life Science. Please subscribe my channel and press the bell icon so you get notifications. Share my video and like it. In this lecture we are discussing about the glycolysis. Before that, the basic terms related with the metabolism. The metabolism is a sum of all the chemical reactions occurring in a cell. It is sum of all the chemical reactions occurring in a cell, and the chemical compounds which involved into the metabolism are called as metabolites. The metabolism is subdivided into two reactions, that is catabolism and anabolism. For a metabolic reaction there is a need of enzymes, and the enzymes plays an important role in a stepwise manner which transports a substrate into a end product. The metabolic pathways can be of following types. It may be linear, for example glycolysis, or a EMP pathway. It may be cyclic like a citric acid cycle, or it may be spiral like biosynthesis of fatty acids. The metabolic pathway serves two functions. It generates the energy, and because of this energy the cell can drive its vital functions. Also, because of a metabolic pathway there is a synthesis of biological molecules, for example production of or synthesis of nucleotides. The metabolic pathway, metabolism means it is a sum of all the chemical reactions occurring in a cell. It is subdivided into a catabolism and a anabolism. The catabolism, in a catabolism there is a oxidative breakdown of a larger complex molecules. Here the larger complex molecules such as carbohydrate, lipids and the proteins get break down into their monomers, and so because of this oxidative breakdown there is a release of energy, and these reactions are called as exergonic reactions where there is a generation of energy. So these are a catabolic pathway where energy is generated. Anabolic pathway or the anabolic reactions. In this anabolic reactions there is a synthesis of compounds, and for synthesis of a compound there is a need of energy, so these reactions are endergonic, and to perform endergonic reaction the energy or a need of energy is fulfilled by a energy which is generated from exergonic reactions. To couple this exergonic reaction and endergonic reaction there is a important role of high energy compounds, for example ATP. Overall, the catabolism is a energy generating pathway, whereas anabolism is a energy requiring pathway of a metabolism. Now, characteristics of the metabolism. Metabolic pathway are irreversible. Every metabolic pathway has committed a initial step. All metabolic pathway are regulated, and this regulation is done by enzymes. Metabolic pathway in a eukaryotic cell occurs in a specific cellular locations. For example, the Krebs cycle or a TCA cycle. In case of eukaryotic cell it occurs in a mitochondria. Now about the glycolysis. The word glycolysis is derived from two words, glycis and lysis. Glycis means a sweet and lysis means a breakdown or a splitting. Here there is a breakdown of a sweet molecule or a breakdown of a glucose. This pathway is also called as Embden, Meyerhof and Farnas pathway. These are a name of scientists who invented the glycolysis pathway. So in short, this is called as a EMP pathway also. Introduction. The glycolysis is a sequence of 10 enzyme catalyzed reactions. Overall, in the glycolysis, one molecule of a glucose get converted into a two molecule of a pyruvate, where there is a production of a ATP. In this oxidative process, one molecule of glucose is partially oxidized to 2 moles of pyruvate. And this glycolysis occurs in a cytoplasm of a cell. The glycolysis occurs aerobically as well as anaerobically, and even though there is not involvement of a molecule oxygen. Once from a glucose there is a production of a pyruvate. In some reaction, the pyruvate get converted into a lactic acid. The glycolytic sequence of a reaction differ from a species to species, and its regulation is also different in a species to species. But in all the organisms, there is a formation of pyruvate, and this pyruvate further get converted into a number of other organic acids, and it depends upon species to species. In aerobic organism, glycolysis is occurs in a cytoplasm, and after a glycolysis it is prelude to a citric acid cycle and etc. That means a glycolysis is a central pathway for a glucose catabolism. Now, here is a fate of glucose. Glucose get stored in form of glycogen in animals, or it gets stored in form of starch in a plant. It also gets stored as a sucrose. Means glycogen, starch and sucrose, these are the storage form of a glucose. Glucose get oxidized in a glycolysis to the pyruvic acid or a pyruvate. Glucose get oxidized via PP pathway or a pentose phosphate pathway into ribose 5-phosphate, and this ribose 5-phosphate is essential precursor for a production of or a synthesis of a nucleic acid. The glucose is a monomer, and it leads to formation of a number of polymers or a polysaccharides which get imparted into a cell wall or a extracellular matrix. So the number of monomers of a glucose get polymerized into a polysaccharide and stored into a cell wall or a extracellular matrix. This is a EMP pathway. 9 reactions are there. 9 to 10 reactions are there where one glucose molecule get converted into a pyruvic acid. Glucose get converted into a pyruvic acid. In first step, glucose get converted into a glucose 6 phosphate with the help of enzyme hexokinase. Glucose 6 phosphate get converted into a fructose 6 phosphate with the help of phosphofructokinase enzyme. Further, the fructose 6 phosphate get converted into a fructose 1,6 diphosphate. Fructose 1,6 diphosphate split into two similar compounds, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate with the help of enzyme aldolase. These two compounds, glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, these are interconvertible compounds and they can be interconverted by help of phosphotriose isomerase. Further, the glyceraldehyde 3 phosphate continues a pathway and it get converted into a 1,3-diphosphoglycerate. Name of enzyme is a triose phosphate dehydrogenase. The 1,3-diphosphoglycerate get converted into a 3-phosphoglyceric acid with the help of enzyme phosphoglyceric acid kinase. 3-phosphoglyceric acid further get converted into a 2-phosphoglycerate. These two compounds are interconvertible or isomers of each other. The enzyme mutase plays an important role for this isomerization. 2-phosphoglycerate further converted into a phosphoenol pyruvate. Name of enzyme for this reaction is enolase. Phosphoenol pyruvate get converted into a pyruvic acid with the help of enzyme pyruvate kinase. This is a pathway or this is a glycolytic pathway. Now in detail, in a glycolysis there are two phases, phase first and phase second. Glycolysis leads to breakdown of a 6 carbon containing glucose into 2 molecules of 3 carbon containing pyruvate acid. Number of enzymes are playing a role for this conversion, and these reactions are bifurcated into two phases. Phase first is called as a preparatory phase and phase second is called as a payoff phase. Preparatory phase, it consists of first 5 steps of a glycolysis in which glucose is enzymatically phosphorylated by ATP to yield fructose 1,6 diphosphate, and this fructose 1,6 diphosphate is then split in 2 molecules of 3 carbon containing glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, these two compounds are 3 carbon containing compounds. Thus, the first phase results in a cleavage of a hexose chain, and this cleavage requires an investment of 2 ATP molecules, and this investment of 2 ATP molecule activate the glucose, and glucose is prepared for its cleavage, and hence it get converted or cleaved into 3 carbon compounds, 3 carbon containing compound. These are the 5 steps, or these are the 5 steps in a phase first or a preparatory phase. Glucose get converted into a glucose 6 phosphate with the help of enzyme hexokinase. Here 1 ATP molecule get breaked and leads to formation of a inorganic phosphate and a ADP molecule. The one inorganic phosphate get attached or get bind to the carbon atom number 6 of a glucose, and glucose get converted into a glucose 6 phosphate. The enzyme hexokinase is playing a role for this conversion, and requires a manganese for its activity. Glucose 6 phosphate, this compound further get converted into a fructose 6 phosphate, as the glucose and fructose, these are isomers of each other. Also, these two compounds, glucose 6 phosphate and a fructose 6 phosphate, are isomers of each other, and this isomerization is carried out by an enzyme phosphohexoisomerase, and magnesium is requiring for the activity of this enzyme phosphohexoisomerase. The fructose 6 phosphate get phosphorylated to the fructose 1,6 diphosphate. Here, one ATP molecule get hydrolyzed into a one inorganic phosphate and a ADP molecule, and that one inorganic phosphate get attached to carbon atom number first of a fructose 6 phosphate molecule, and leads to formation of fructose 1,6 diphosphate. The name of enzyme for this reaction is a phosphofructokinase. The fructose 1,6 diphosphate is a 6 carbon containing compound and 2 phosphate containing compound. This compound further split into a two similar compounds, that is dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. These two compounds contains 3 carbon atoms and 1 phosphate molecule each. And these two compounds, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, are isomers of each other, or these are interconvertible. The phosphose triose isomerase can interconvert these two compounds. For synthesis of these two compounds from fructose 1,6 diphosphate, aldolase plays an important role. Then, payoff phase. This phase constitutes the last 5 reactions in a glycolysis, and in this phase there is the actual production of ATP molecules during the conversion of glyceraldehyde 3 phosphate to the 2 moles of pyruvate. Here 4 moles of ADP are phosphorylated to the ATP and leads to formation of 4 ATP molecules. But actually, in a preparatory phase, there is a consumption of 2 ATP molecules, and here in a payoff phase, there is a generation of 4 ATP molecules. So 4 minus 2, here the net result is a 2. That means when 1 mole of glucose is oxidized, and it leads to formation of 2 moles of ATP. Here is these are the reactions in payoff phase. Once the glyceraldehyde 3 phosphate is formed from splitting of fructose 1,6 diphosphate, this glyceraldehyde 3 phosphate get dehydrogenated and phosphorylated with the help of enzyme triose phosphate dehydrogenase, and hydrogen molecule get released from the 3 glyceraldehyde 3 phosphate. This released hydrogen molecule is accepted by a NAD, and the NAD is converted into a NADH2. Here is a impartment of or incorporation of one phosphate molecule in this glyceraldehyde 3 phosphate, and hence it leads to formation of 1,3-diphosphoglycerate. Overall, in this reaction, there is a utilization of one phosphate molecule and formation of NADH2. Once the 1,3-diphosphoglycerate is formed, it get dephosphorylated. And because of this dephosphorylation, the rigid phosphate molecule is accepted by one ADP molecule and forms a ATP, and this is an example of a substrate level phosphorylation. 1,3-diphosphoglycerate get converted into a 3-phosphoglycerate. Here is a production of a ATP molecule, and this is a substrate level phosphorylation. The enzyme, that is a phosphoglyceric acid kinase, and this reaction is a reversible reaction, and for activity of phosphoglyceric acid kinase enzyme, magnesium ions are necessary. The 3-phosphoglyceric acid get isomerized into a 2-phosphoglycerate. These two compounds are having a same molecular formula, but they show a different structural formula. Here the phosphate group is to the carbon atom number 3. Instead of it, in a 2-phosphoglycerate there is a phosphate to the carbon atom number 2. So these are different in their structure only. They possess same molecular formula. And this reaction is also reversible, and this is a isomerization reaction. The enzyme, that is a phosphoglycerate mutase, plays a role for this conversion. The 2-phosphoglyceric acid get dehydrated to the phosphoenol pyruvate. The enzyme for this reaction is a enolase, and once the phosphoenol pyruvate get formed, a phosphate molecule formed phosphoenol pyruvate get released, and it is accepted by a ADP molecule and forms a ATP. And this is a second example of substrate level phosphorylation, and the enzyme for this reaction is a pyruvate kinase. From dephosphorylation of phosphoenol pyruvate, there is a production of a pyruvic acid and a ATP molecule. This reaction is an example of a substrate level phosphorylation. Now we can see the stepwise explanation of a glycolysis. Step first, that is a phosphorylation. In the phosphorylation, the glucose molecule is phosphorylated by a ATP. Here the ATP molecule get hydrolyzed and it forms a ADP and one inorganic phosphate. This one inorganic phosphate is accepted by a glucose, and glucose get converted into a glucose 6 phosphate. The name of enzyme is a hexokinase. This is irreversible reaction, and this reaction can be represented as like this. Step second, isomerization. It is a reversible step, and here there is a rearrangement of a chemical structure of a carbonyl oxygen form, carbon atom number first to second, and here ketose form get converted into a aldose 1. This isomerization of aldose 6 phosphate gives ketose fructose 6 phosphate. This is a ketose and this is a aldose. Glucose 6 phosphate is a aldose and fructose 6 phosphate is a ketose. And these are the isomers of each other. They having a same molecular formula but different structural formula. And the enzyme for this conversion is a phosphoglucoisomerase. This reaction is reversible. Means these two compounds are interconvertible. Third step, phosphorylation once again. Here, fructose 6 phosphate molecule is phosphorylated by a ATP to a fructose 1,6 diphosphate. In this reaction, ATP molecule get break down into a ADP and inorganic phosphate. The inorganic phosphate get attached to the carbon atom number first of fructose 6 phosphate, and fructose 6 phosphate converted into a fructose 1,6 diphosphate. The enzyme for this reaction is a phosphofructokinase, and this reaction is irreversible. Fourth step, breakdown. The 6 carbon containing compound or a molecule, that is a fructose 1,6 diphosphate, get split or get cleaved into two 3 carbon containing molecules, that is glycerol 3 phosphate, that is GAP, and dihydroxyation phosphate. These two compounds contain 3 carbon each and 1 phosphate each. So these two compounds, glycerol 3 phosphate and dihydroxyation phosphate, these are isomers and interconvertible molecules, and this interconvertible by enzyme triose phosphate isomerase. But for a splitting of fructose 1,6 diphosphate into glycerol 3 phosphate and dihydroxyation phosphate, aldolase plays an important role, or the reaction is catalyzed by aldolase. Then next step, that is step 5, isomerization. After splitting of fructose 1,6 diphosphate into glycerol 3 phosphate and dihydroxyation phosphate, the triose phosphate isomerase can interconvert the dihydroxyation phosphate to glycerol 3 phosphate or vice versa. Out of these two, glycerol 3 phosphate continues to the glycolysis, and also this remaining compound dihydroxyation phosphate further get converted into a glycerol 3 phosphate and continues a glycolysis. We can see that, or we can say that there is a formation of two molecules of glycerol 3 phosphate. Step 6, two molecules of glycerol 3 phosphate are oxidized with the help of enzyme glycerol 3 phosphate dehydrogenase, and during this conversion, the released hydrogen molecule from glycerol 3 phosphate is accepted by NAD and forms a NADH2, and glycerol 3 phosphate converted into a 1,3 phosphoglycerate. Two molecules of glycerol 3 phosphate get converted into a two molecules of 1,3-diphosphoglycerate. Enzyme for this reaction is a glycerol 3 phosphate dehydrogenase, and in this reaction, two NADH molecules are formed and two ATP are formed. This reaction is a reversible reaction. The next step 7. In step 7, here is a transfer of a high energy phosphate group to form a ATP, and the molecule, that is a 1,3-diphosphoglycerate, get dephosphorylated and leads to formation of a 3-phosphoglycerate, and this is a substrate level phosphorylation where there is a formation of a ATP molecule. The molecule, that is a 1,3-diphosphoglycerate, a 1-phosphate molecule, is accepted by a ADP, and this ADP get phosphorylated to a ATP. So this is an example of substrate level phosphorylation. Here the phosphate donor is substrated a 1,3-diphosphoglycerate. In step 7, the 1,3-diphosphoglycerate get dephosphorylated to the 3-phosphoglycerate. Once 3-phosphoglycerate molecules are formed, there is the release of 2 NADH and there is a release of 2 ATP molecules. Then step 8, the remaining phosphate ester linkage in 3-phosphoglycerate is moved from carbon atom number 3 to 2. This is also a isomerization. The reaction is catalyzed by a phosphoglycerate mutase, and the molecule of 2-phosphoglycerate are produced. This is also a isomerization reaction catalyzed by a phosphoglycerate mutase. In this reaction only the position of phosphate molecule get changed. Here phosphate molecule is at carbon atom number 3rd. Here the phosphate molecule is at carbon atom number 2. So these are the isomers of each other, and this is a reversible reaction. Then next step 8, dehydration of 2-phosphoglycerate, removal of the water molecule from 2-phosphoglycerate. This is a second reaction in a glycolysis where high energy phosphate is formed. In this reaction, the 2-phosphoglycerate is dehydrated by the action of enzyme enolase, and phosphoenol pyruvate is formed. This compound is a phosphate ester of a enol or a enol tautomer of a pyruvate, and this conversion is a reversible. Two phosphoglycerate get dehydrated to a phosphoenol pyruvate. Reaction is catalyzed by a enolase. This is a reversible reaction. Then step 10, transfer of phosphate from phosphoenol pyruvate to a ADP. This is a last step and it is irreversible. Here phosphate group from phosphoenol pyruvate is transferred to a ADP, and ADP get converted into a ATP, or this is a phosphorylation reaction catalyzed by an enzyme pyruvate kinase, and this is a second substrate level phosphorylation, and here is a actual yield of a ATP molecule, and this is a non-oxidative phosphorylation reaction. Phosphoenol pyruvate get converted into pyruvate, dephosphorylation of a PEP. Because of this dephosphorylation, rigid phosphate molecules are accepted by a 2 ADP molecule and leads to formation of 2 ATP molecules. This is a second example of substrate level phosphorylation, The reaction is catalyzed by a pyruvate kinase. Now, overall balance sheet of a glycolysis. In a glycolysis, one glucose molecule gives a two molecule of glycerol 3 phosphate. Therefore, the total input obtained reaction can be as glucose plus 2 ATP plus 2 inorganic phosphate plus 2 NAD molecules plus 2 hydrogen plus 4 ADP gives 2 pyruvic acids, 2 hydrogen molecules, 4 ATP molecules, 4 water molecules, sorry, 2 water molecules, 2 NADH and 2 ADP. This is a balance sheet of a glycolysis. If we cancels a common terms from above equation, we get a net equation for glycolysis as glucose plus 2 inorganic phosphate plus 2 ADP plus 2 NAD leads to formation of 2 pyruvic acid molecule, 2 NADH, 2 ATP, release of 2 hydrogen and 2 water molecules. Thus, the simultaneous reaction involved in a glycolysis are: it is not only a ATP generation process, it also leads to formation of a reducing power in form of NADH. Here, the glucose is oxidized to a pyruvic acid. NAD is reduced to a NADH. That means it leads to formation of a reducing power. ADP is phosphorylated to a ATP, which release a water molecule. Now, a energy yield in a glycolysis. In a payoff phase or a first step, there is a consumption of one ATP molecule, where a glucose get converted into a glucose 6 phosphate, and the ATP is break down into a ADP and inorganic phosphate. So there is a consumption of one ATP. In a third step, fructose 6 phosphate get converted into a fructose 1,6 diphosphate. Here, a phosphorylation process is there and breakdown of ATP. So consumption of one ATP is here. At a step number 7, where 1,3-diphosphoglycerate get converted into a 2-phosphoglycerate, and this is the first example of a substrate level phosphorylation. The released phosphate molecule are accepted by a 2 ADP molecule and forms 2 ATP molecules. So the gain of ATP in the step number 7 is a 2. Step number 10, it is also a substrate level phosphorylation. Phosphoenol pyruvate get dephosphorylated to a pyruvate molecule, and the released phosphate molecule is accepted by a ADP and leads to formation of ATP. The number of ATP formed in this reaction are 2. So net gain of ATP is 4, consumption of ATP is 2. 4 minus 2, net gain of ATP is 2. So the glycolysis, in a glycolysis, the net gain of ATP is a 2. Thank you. If you like my video, please subscribe my channel. Press the bell icon so that you will get the notifications. Share my video to your friend, like, and if I have a mistakes, please tell it me in comment. Thank you once again.